Here to talk about the state of vision research today in Canada is Dr. Bob Gendron. Thank you, Bob.
Thank you very much, Sharon. Can everybody hear me okay? Everybody in the back? Yeah, excellent. Well, thank you all again for coming. This has been fantastic. This has been going on for a couple days now. And as I said yesterday to some of my colleagues and some of the people that are attending this meeting, it's a real pleasure. It's actually a rare pleasure for us to get together with people that are dealing with vision loss. So this is the kind of thing that we look forward to as researchers. And I just wanted to say that we're not going to be using any slides, and that all these talks will be recorded and will be posted on ffb.ca, so you can go online and go back to them if you want more information. So I thought I'd start by saying that it's maybe I hate to say it's about 2 decades since I started and since I graduated with my PhD at McGill. And looking back, I think one thing really strikes me when I look at vision research today across the world, and that including Canada, and that's how the amazing use of knowledge applies to things like treating and finding cures for retinal disease. We see other fields helping us in learning about the retina and also in how we can treat the retina. And back when I started, I'm studying AMD now, age-related macular degeneration, but back when I started, it was about 10 or 15 years ago, there were no treatments for wet AMD. So the wet AMD, as I'll be talking about later, is the form of the disease where you get blood vessel overgrowth. And back then, there were no treatments for it. And it's incredible because now with the field of cancer, we've learned from the field of cancer that you can actually derive these drugs that are directed against blood vessels that are central to cancers. And although those drugs are not really useful in cancer, they've been clinically tried, they're not all that effective, but they're effective in retinal disease. So to me, that's really incredible, and it's exciting. Again, an example of how another field can affect something like retinal disease, and we get drugs that may not work in one disease, but they're very effective in another disease like retinal disease. So you'll see a lot of that, I think, in the talks today, where you have knowledge that's shared from one field to another. And we're talking about cutting-edge research today, and I have the privilege of being a member of the Scientific Advisory Board for Foundation Fighting Blindness. So in those years that I don't have a grant that's submitted to the panel, since we review our panels without the people whose grants are in them being on the panel, I'm actually sitting on that panel and looking at some of the most cutting-edge research that's occurring in Canada. And by doing that, I'm actually seeing and reading about a lot of the research across the world and how Canada relates to that research. Because when people write grants, they're incorporating knowledge from around the world. So it's really a world-based type of project that you're looking at being done here in Canada and funded by the Foundation Fighting Blindness, which is very important. And today what I'd like to do is share with you some of those projects that are going on. And really, by no means all of them. These are just a few snippets of information about the projects that Foundation Fighting Blindness funds and that are going on across Canada, not only in our labs in St. John's, but across Canada, from Vancouver to Alberta to Toronto to Montreal, and of course Halifax. So what we do know now, and that, again, incredible, is that the retinal diseases that include both the childhood retinal dystrophies as well as the age-related macular degenerations involve a huge number of genes. And right now I can tell you that there are probably even more than this, but I think that there's about 250 genes that are somehow mutated or changed in some way that are involved in these retinal diseases from childhood to aging. And for me, that's incredible, 250 genes. So now we know we're getting closer to what those genes do and how they can be possibly used for treating retinal disease. And if we couple the knowledge of those genes, those 250 genes, with as I mentioned earlier, the knowledge that we're getting from other fields, we can then start applying that knowledge to new treatments. And one example of that is what we call gene therapy. And I'm sure many of you have heard about gene therapy, and you're probably excited about it, and we are as well. And that's really in its infancy now. In the last 10 years, gene therapy has been coming along and moving slowly through clinical trials and then getting applied to even things like retinal disease. So just as an example for that, There's a disease called Leber's congenital amaurosis, which is a very severe retinal dystrophy that occurs in early childhood in some individuals because of a number of gene mutations or gene changes in the body. And over the last 10 years or so, there's again been an incredible marriage of knowledge, both from the retinal disease field, where we're trying to understand what the genes are that cause this disease, and also fields like virology, where people are taking viruses that are actually within us in the body, and these viruses are normal. Many of them are non-pathologic. They don't cause disease, but when we take these viruses and study them, we can use them kind of like a Trojan horse, but a good kind of Trojan horse, all right? So they're actually used to re-engineer the normal gene. In this case, it would be the normal Leber's congenital amaurosis gene. And then that virus is used to then implant it back into the retina to try to make those retinal cells that are missing that gene to express the normal gene. And that's been done now in a very limited clinical trial in the US and the UK, and a very limited number of patients, but there were actually very surprising and exciting results from that trial. And those trials are ongoing. And that occurred when a gene called RPE65, which produces a protein that's important for the photoreceptor health and well-being, when that gene is replaced with the viral vector, with the normal gene, these individuals that received that treatment actually had some residual vision reoccurring. So they actually got some of their vision back after being completely without vision. So that's extremely exciting. The problem with those types of approaches, and we all deal with it, is that they're very slow. So gene therapy occurs over very many years, and it has to, because we need to test these therapies both for safety in phase 1 trials, and then efficacy and safety in phase 2 and phase 3 trials, where we increase the number of patients that are in those trials. So there's hope there. There's trials ongoing for that disease using gene therapy, and gene therapy is being applied to many other retinal diseases. And if you go online to clinicaltrials.gov.gov, you can actually see, or for some of you hear, all of those clinical trials that are going on in the US and the UK. And it's incredible to see how many of those trials are going on that are directed to retinal disease. So getting back to the science, again, the other thing that really excites me and excites some of us in the field is sometimes the simple benefits of doing basic genetic research. And as Jane Green will talk to you later, there are many genes out there. Some of them will have to be dealt with through gene therapy, but there's others that can serendipitously be dealt with through very simple means, but we have to understand what those genes do. And another example is, again, the Leber's congenital amaurosis disease. And there's a doctor in Montreal, Dr. Robert Koenig, who's also funded by the Foundation Fighting Blindness, and he's right now leading a trial for a drug called QLT09. And this drug was derived because of our knowledge of the Leber's congenital amaurosis genes like RPE65. And it's known that that gene creates a chemical in the eye, a vitamin-like chemical, that then is not present in these Leber's patients. And what Dr. Koenig is working on is a synthetic analog of that chemical. It's almost like a vitamin. It's kind of like a form of vitamin A. And when these patients are given this chemical, the QLT09, they're actually regaining, again, some of their health of their photoreceptors and their vision. And so that, again, that trial is ongoing. It was reported last year at ARVO, the Association for Research in Vision and Ophthalmology, and that's a very exciting new approach to try to rescue the health of those photoreceptors using that chemical. The gene discovery and gene therapy are not really the only new and exciting approaches that scientists across the world and across Canada are using. And another example of some of the incredibly interesting approaches and exciting approaches are stem cells. And you probably, again, heard of some of the use of stem cells, and we'll be hearing more about stem cells and progenitor cells later today when Dr. Caillouette, our other visitor from Montreal, will be speaking about them. But many years have passed since the early 80s when scientists first realized that you can take a stem cell. Back then it was from an embryo, mainly mice embryos, and if you take these stem cells out of the embryo and put them in a dish, they will differentiate or change into any tissue. And that includes retinal tissue. So in the last decade or so, people have been studying how these stem cells can be coaxed in vitro to create retinal tissue. And that's extremely exciting. And again, I'd like to give you just a bit of information on a trial, that, again, another clinical trial that's being done in the US right now that's using stem cell-derived retinal pigment epithelium. And as I'll be talking about later in the breakout session, you probably also know that the retinal pigment epithelium, which is the layer of cells that lies right behind the photoreceptors, is central to the health of the photoreceptors. And what this trial is doing is taking stem cells that are actually from human embryos, and now it can be done from tissue that's actually from adults. There's technology to differentiate tissues from adults into stem cells. They're taking these cells and implanting them after they've been differentiated into retinal pigment epithelium back into the patient's retinas. And they're getting some preliminary results that the trial is safe, so there's been no adverse effects. And they're also getting some very preliminary results of efficacy. So that means that the treatment is actually thought to be effective. So what this allows us to think of in the future is that someday, maybe using adult stem cells, someone can maybe take a patient's cells, maybe from their skin or from another part of their body, and create retinal tissue from them. So that's, again, far off. This is not the type of thing that's going to be very soon, but it's probably 10 years into the future. So there is hope there, though. And you'll hear more about that from Dr. Cayouette. And there are other people in Canada that are funded by the Foundation Fighting Blindness that are using stem cells. So there's many other approaches that I wanted to talk about. I'm clearly running out of time here, and maybe I'll talk about them more at the breakout session, as well as Dr. Green and Dr. Cayouette. But I wanted to end that saying that this is truly, vision research and retinal research is truly a quest. Like today, we have Vision Quest, and the most important part of that quest is all of you. You're the central part of this quest. That's why we're here. That's why we work in our labs. That's why the foundation exists. We want to try to understand and cure retinal disease. And a lot of other people are involved here. Your optometrist, your ophthalmologist, your primary care physician, those are the people that are the first line that can maybe detect early retinal disease. Your local CNIB, they offer resources for vision loss. So all these people are part of the quest. Your neighbor down the street that rides a motorcycle, the Thunder Rider wearing the leather jacket, that person might be going out and getting pledges for Foundation Fighting Blindness, Ride for Sight, which is a fantastic thing, and I just can't say enough about it. It's great. So all these people here, including people like us, like Yelena and I, that work in our lab over at MUN, our lab is working on macular degeneration, mainly the wet form, and we're interested in trying to learn how the blood vessels in that disease leak. And they leak fluid, they leak all kinds of materials into the retina and then damage the retina. So we're working on a protein that we think is involved in stabilizing those retinal blood vessels that are leaking. And so we think that our potential treatment might be used in combination with other treatments that are available today, like the anti-VEGF therapies. So it's been a real trajectory. Leber's congenital amaurosis was found in the 1800s. The virus that I told you about was determined or found out in 1965, and the treatment occurred, or the first trials occurred in 2007. So that's 140 years. Okay, that's a long time. But I'd like to tell you that if you look at other fields, and I take again cancer as an example, we have childhood leukemias now, the genes of which have been characterized only in the last 20 years, and now, if you're a child with leukemia, you have an 85% chance of being cured of your leukemia. Okay? Another example is a drug called Gleevec. That drug was derived across about a decade starting in the late 90s, and now it's used to treat over 10 different cancers. And that's why I'm excited about retinal research, because I think the same thing is now starting to happen in an exponential fashion, like a snowball, to try to address retinal disease. And there are not as many of us doing retinal disease research as there are cancer research, but we're here, we're in Canada, we work with the rest of the world, and the Foundation Fighting Blindness funds us and supports us, and that's why we're excited. So I hope today you find the conference interesting, and please ask questions. I'm not sure I can answer all of them. I probably can't answer half of them, but I'll try my best, and we hope you enjoy your day and you get the most out of it you can. So I'll end there. I think hopefully I'm on time, and we'll close. Thank you very much. No questions? It was that clear? Oh, maybe there's one here.
Dr. Gendron,
Yes.
you mentioned gene therapy, and we've been hearing a lot about gene therapy over the last number of years. I'm just wondering, in terms of clients for gene therapy, like, is this something that's seen as being effective for all different forms of retinal diseases? Is there one client group who it's more beneficial for in terms of ages or different types of diseases? Or do you see it as an umbrella type approach to...
It's not an umbrella type approach, and one good example of that is in age-related macular degeneration. Because in that disease, and I'll be talking about this more later, there's a multiple number of genes that could be involved in a multifactorial way in that disease. And not only that, in that disease there are a number of environmental factors that occur. So there's smoking, there's diet, there's lifestyle, and all those converge onto a disease spectrum that's different for every person. So gene therapy for late-stage AMD, either dry or wet, is probably not the optimal treatment. The gene therapies are probably going to be best for those individuals that might have a very specific mutation or change in one gene, where you can go in, like they did in the Lieber's case, and correct that gene mistake, and then create either a new healthy protein that gene makes that then nourishes the retina, or will change the tissue in some way that's very specific so that the gene therapy itself is not toxic to the rest of the retina or the rest of the body. So I hope I answered your question. Yeah. Yes?
I have Stargardt's disease, and I noticed you didn't mention it this morning, but I'm wondering what's happening.
Oh, maybe you could use the microphone if you don't mind. Thank you.
Like this?
Thank you.
Hi, I have Stargardt's disease, and
Right.
I noticed you didn't mention that this morning, and I'm assuming that it's included in the topics that you discussed.
Yes.
But I'm wondering, with Stargardt's, what's happening right now? Do you have any information about that?
Yeah. Maybe Jane would be best to answer that question. She's our local expert on these types of diseases, so we have a mic for Jane?
In the next breakout session, where I'll be talking about some of these hereditary conditions, and Stargardt's disease is one of the hereditary conditions. And so I also will be, talking about what the different approaches that are being taken now with Stargardt disease, because that's actually one of the fairly common, there's many different hereditary diseases, but that's one quite significant one. And so, that's one of the things I will be talking about in the next sort of session. So yes, there's things happening with Stargardt disease. There's several different types of approaches that people are using to try and help out.
The other interesting thing about Stargardt's is that some of the forms of Stargardt's share similarities with some of the macular degenerations. And so I think what you'll be seeing is that as we learn more about how to treat, especially some of the dry AMDs, that the approaches are going to kind of bleed into the other diseases like Stargardt's. And it all revolves around retinal health. So if you maintain the retina in a healthy state, and that involves diet, supplements, and lifestyle, that all that converges, as I said to this other gentleman, into a kind of an overall more rounded treatment to these specific diseases. So there's a lot of application from one disease to another. So.
Dr. Gendron, would you tell us a little bit about Psyop therapy?
Oh, sure, yeah. The Psyop is a gene that's involved in regulating how retinal cells die or live. So there's actually an interesting process in all of our tissues in our body called apoptosis, where we have programmed cell death. So the cell it's almost like the cell has a little computer inside it that tells it a program to go through where it goes to death. And that sometimes happens in some of these retinal diseases. So there's a program for some abnormal reason for some of those photoreceptors or retinal pigment epithelial cells or retinal neurons to die. And there's a very exciting project that's being led by Catherine Silfitis from Ontario, who's also funded by the Foundation Fighting Blindness. And she's trying to figure out, again, a gene therapy using this Psyop protein or gene that seems to prevent or block this cell death that we're seeing and this programmed cell death. So her work is actually moving now toward into preclinical modeling, where we're actually testing the potential treatments in mice. Some of these mice are highly engineered to be genetically different. And her particular work is going through the preclinical stages, and in the next, again, 5 to 10 years, hopefully we'll go into clinical trials in humans. Again, targeting the Psyop protein into the retina to try to save those cells from dying. So, again, very exciting.